Hello everyone. You are looking at Pennycress fields in St. Paul campus. These fields represent mutation breeding and trait discovery program here at the University of Minnesota. These Pennycress plants were planted in a 4x4 plots and these were derived through mutagenesis. These lines represents variation for several chemical attributes such as oil content, protein content, glucosinolate content, and fatty acid variation. Along with the seed attributes, we also look for variation in plant development, such as plant earliness, plant height, plant maturity, number of tillers, and something that I can right now point you guys to is the differences in plant height that we observe in this mutant population, and that has been heritable for several generations. As you can see, most of these plots represent a tall pennycress, and what I'm zooming to is a tall pennycress wild type plant compared to these dwarf plants that you see here, which are significantly shorter. And moving on to the trait discovery efforts, what we've been doing is we've been selecting M2 plants and subjecting them to whole genome sequencing and then harvesting the seeds and planting them in the rows. As you can see here, that we have these 4 rows and each of these rows represent a different M2 plant. And then we look at the whole genome sequence data and tag about 12 plants in each plot. Those orange tags are meant to allow us to isolate DNA. We isolate DNA from these 12 plants, refer back to the genomic data so that we can isolate homozygous mutants. This summer, we have planted over 100 M2 lines, and our hope is to isolate at least 50 homozygous mutants in 50 different genes that would allow us to continue our Pennycress improvement program, and these mutations would represent several different developmental traits. Using that mutant population, what we've been able to do is successfully identify mutations that are useful for seed chemistry or agronomics. And this plot here represents the variation that we were able to combine through CASP markers and find this triple mutant, which is high oleic and has a reduced shatter phenotype.